Hello, here's my lecture notes for Chapter 6, Metabolism, Energy, and Enzymes. So the definition of energy is the ability to do work or bring about change. And all living things need a constant supply of energy to survive. And energy allows us to grow, respond to stimuli, metabolize things, and reproduce. Just remember, life on Earth is dependent on solar energy, and photosynthesis provides nutrients for the majority of organisms on Earth. So very important, energy comes in two forms, at least, of kinetic energy, which is the energy of motion, so when a ball rolls down the hill, and there's also potential energy, which is stored energy. So food, for example, has stored energy. So you could think of types of energy as well. There's chemical energy stored in chemical bonds, and mechanical energy is energy of the motion of walking. So there's two laws of thermodynamics, and just remember that energy flows through an ecosystem. So for example, on Earth, photosynthesis is where plants capture solar energy and store it in chemical bonds of carbohydrates and sugars. Plants use carbohydrates in cellular respiration, just like animal cells, and plants serve as food for other organisms. And just remember, energy is lost as heat in every step. So, very important, the first law of thermodynamics states that energy cannot be created or destroyed, but is changed from one form to another. This is the law of conservation of energy. And the second law of thermodynamics states that energy cannot be changed from one form to another without loss of usable energy, usually in the form of heat. So here we have sun going into a chloroplast in a plant. Carbohydrate synthesis occurs, CO2 and water go in, and heat and O2 are released. So here we have muscle contraction in our muscle cells. So we use carbohydrates to burn and ATP to put this into mechanical energy so we can move. And remember, some of the energy is lost as heat. So if you've ever run long distances, your whole body gets hot and sweats. That's due to all the ATP we're using. All right, so the heat released from cellular respiration or ATP usage dissipates into the environment. All right, and the heat generally cannot be captured and converted into more useful forms. So this one's kind of conceptually hard to digest, I think, but there is a concept called entropy, and it refers to the amount of disorganization in the world, in a cell, in your room, in your my lecture notes, and the strange thing is that you would think we want to go from an unordered to ordered world, but it's actually the opposite. So every process that occurs in a cell increases the total entropy or unorder of the universe. And this is a difficult concept to digest, so I'm going to skip ahead to a couple slides where I think it really kind of hits home. So there it is in the chemical world. There it is in the channel world. Here it is in the real world. So everyone, well, at least most people I would think, would want to have the bedroom on the left. But in reality, I think most of us live with something that's a little more on the right. Okay? So metabolism is the sum of all chemical reactions in the cell, and it's broken up into two parts. Catabolism is the breaking down of molecules, and anabolism is the building of molecules. Remember, in a chemical reaction, the reactants are on the left, A plus B, and the products are on the right, it's C plus D. Okay. And for a reaction to go, we have to look at the free energy that's around. That's called a delta G. Delta means change. And a negative delta G means the reaction will go forward and proceed. So exergonic reactions are spontaneous and release energy, and endergonic reactions require an input of energy. So very, very important in the biology world, ATP is the energy for cells. It's what keeps us all alive. It's the common currency, and it stands for adenosine triphosphate. So ATP is generated from ADP, adenosine diphosphate, by the addition of an inorganic phosphate molecule. ATP is generated by glucose breakdown, and it can be used to power many reactions in the cell. So the structure of ATP, it's very similar to a nucleotide. So it's adenine, a nitrogen-containing base, a ribose, a 5-carbon sugar, and 3 phosphate groups. And the energy is stored in the chemical bonds between the phosphate groups. So here's just a model of the conversion between ATP and ADP. You can review this on your own. So ATP can be used for chemical work to synthesize macromolecules, transport work to pump substances across membranes, or mechanical work, so muscles can contract, cilia and flagella move, etc. All right, so coupled reactions is the energy released by an exergonic reaction drives an endergonic reaction. So here we can see ATP is converted to ADP, and it requires a little input of energy. All right, so 2 ways to couple ATP hydrolysis to an endergonic reaction is it energizes a reactant, or ATP can change the shape of a reactant. And both can be achieved by phosphorylation or the transfer of a phosphate group to a reactant. All right, so once again, examples of ATP-powered reactions. You can read this on your own. Here we use ATP converted to ADP for muscle contraction through myosin, the protein that's common in your muscles. All right, so metabolic pathways are usually a series of linked reactions, and these are very organized, and they begin with a specific reactant to produce a specific end product. And enzymes are proteins that act as catalysts to speed up chemical reactions. The one exception is a ribozyme. These are RNAs or nucleic acids that can act as catalysts, but 99% of the time, enzymes are proteins. And they participate in the chemical reactions, but are not used up. And neither to determine if the reactions go forward, they're just aids. So here we see reactants of going through a series of pathways. The enzymes, enzyme 1, 2, and 3 above, help it along. So we start off with A, and we end up with product D, as in dog. So the energy of activation, or EA, is the energy that must be added to cause reactions to react with one another. So even in exergonic reactions, where there's a negative delta G, energy of activation must be overcome. So the example is you need to start a match to start burning wood, or I think more simply, you need a match to shoot off a firecracker. It has to start the wick to burn. The enzymes lower the EA, but do not change the end result of the reaction. It just increases the reaction rate. So here's just a graph of looking at what enzymes do to lower the EA. And enzymes usually have an active site, which is like a pocket, where the substrate fits so that they can do their job. So some enzymes might degrade products, and some enzymes might synthesize products. So here's a degradation of reaction graphed out for you, or a model. And here's one where substrates are put together, a synthesis to make a larger product. So enzymes function by what's called an induced fit model, and it's where the substrate fits into a groove in the enzyme or protein, and then the enzyme or protein slightly moves to facilitate the enzymatic reaction. So here's the active site of an enzyme that's colored in blue, and the substrate is in yellow. So here's a list of enzymes. So there's lipases, urases, maltases, ribonucleases, lactases. These enzymes all break things down. So lipases break down lipids, urases break down the compound urea, maltase breaks down the sugar maltose, ribonucleases break down RNA or ribonucleic acid, and lactases break down the sugar lactose. So if you notice, there's a common pattern with enzymes. They generally end in ASE. So these are different things that affect enzymatic speed. Okay. So substrate concentration, the higher the substrate, the faster the reaction can go. Temperature and pH can also affect enzymatic activity. Enzyme activation and inhibition plus cofactors. So some enzymes need friends to help the reaction along. So here you can see substrate concentration. If there's more enzyme and substrate, there'll be more collisions. So more substrate will fill the active site. And the maximum rate is when all the active sites are filled with substrate. Temperature and pH can also affect enzymatic activity. Enzymes have a certain pH they like and a certain temperature. So here are just different examples of how temperature and pH can affect enzymes. Obviously, a polar bear keeps itself warm to keep its enzymes moving. Sometimes body temperature of cold-blooded of animals can slow down their enzymes as they get cold. And there's just a graph of rate of reaction as a function of temperature on the far left. So like I said before, each enzyme has an optimal pH and temperature it prefers. Example is pepsin in the stomach has a low preferred pH because that's where it's made to work. So pepsin, which breaks down proteins, likes a pH around 1 or 2. Trypsin, another protease, likes a pH more around 7 or 8. All right, here's an example of enzyme activation. So kinases add phosphates to proteins that either activate or inactivate them. And so there's also enzyme inhibition, and this can occur when the substrate cannot bind to the site. All right, and almost every enzyme in the cell is regulated by some sort of feedback inhibition. All right, so the simplest type of inhibition is when the product is abundant, it binds the enzyme active site and blocks further production. And then when the product is used up, it is removed from the active site and the enzyme begins to function again. These are more complicated enzymatic inhibitions, so there might be a secondary site that can inhibit it. All right, here's a graph of how enzymatic inhibition occurs. You can review this on your own. So like I said before, a lot of enzymes like cofactors. So a lot of times it's a metal. So copper, zinc, magnesium, manganese can be cofactors. Or there are organic non-protein cofactors called coenzymes, such as NAD+, FAD, NADP+. And vitamins can also be components of cofactors. So in metabolism, we have a lot of oxidation and reduction reactions. So oxidation is a loss of electrons. Reduction is a gain of electrons. And these are usually coupled. So as one of atom or molecule gets reduced and gains electrons, of another molecule or atom becomes oxidized or loses electrons. So the term oxidation, just so you know, is even used when oxygen is not involved. So here is an example. If you put Na plus Cl minus together, you get NaCl. So the sodium is oxidized and the chlorine is reduced. And these terms also apply to covalent reactions involving hydrogen ions. So just so you know, it applies there too. All right, so chloroplasts and photosynthesis. So chloroplasts convert solar energy to ATP and use it to reduce carbon dioxide to carbohydrates. Oxygen is the byproduct that is released. And here is the overall equation for photosynthesis. So we take the solar energy for the sun plus 6 CO2 molecules plus 6 waters, and results in glucose, C6H12O6, and 6 oxygen molecules. So the mitochondria oxidizes carbohydrates and uses the energy to build ATP. In this reaction, oxygen is consumed or used up, and CO2 and water are produced or released. And it's basically the opposite of the photosynthesis equation. So it's glucose plus oxygen releases carbon dioxide plus water plus ATP. All right. So just remember that when we are making or synthesizing ATP as our energy source, we're also using up energy to do this. So here is just a little model or illustration showing that, in plants, sunlight plus CO2 plus water will give us, carbohydrates. We burn those carbohydrates to get ATP and release water and CO2. All right. So, we eat food from plants, or we eat animals that have eaten plants. So we need plants for us to basically make ATP. And so if we do not have plants, we would basically starve to death. And this is the last model or diagram for this chapter. And it shows we breathe in O2 and we exhale CO2. And by eating, we burn our calories through carbohydrates, proteins, and sugars to make ATP in the mitochondria, which gives us energy. And that is all for this chapter. Thank you.